Yeah, really.
Well, good evening, ladies and gentlemen, and a warm welcome from a cool Cape Town. It's been a wonderful day over here, cloudless and no wind. So, if you were here as a visitor, and I'm sure you were, some of you, it's a remarkable day in a beautiful winter. We just had a beautiful SASOG meeting, and that's why there was no webinar last Thursday. And welcome to all those that have been joining us this evening. Our host is Professor Steve Jeffrey, and my name is Peter de Jong, as always. And we have a wonderful speaker tonight, Dr. Tina Wojno, and she is from Lancet Century City. And as you know, she's originally from WITS and UCT, and she'll be presenting vaginal microbiomes and the NGS, and that stands for non-genetic sequencing. So it's been a great pleasure to have her. She's been on this platform before. Just before we start, Chris, our trustworthy cave dancing, cave boxing technician, mentions that some of you might not have got the CPD and the invitations. If that is the case, there is a slight gremlin on our IT system. So please let me know, WhatsApp or email me, and I'll pass on your details to Chris to add on to our database. So without further waste of time, it's wonderful to have you, Tina. As always, you've spoken on this very, very important platform on the very, very important subject of vaginal microbiomes. So vaginal microbiomes, could you start sharing your slides, please?
I shall do that. Thank you so much for the lovely introduction, and thanks for having me again on this platform.
Our pleasure.
Thank you so much. Let me share my slides. Oh, wait, hold on.
The vaginal microbiome and NGS.
Okay, I'm sharing my screen. Can everybody see that?
We can. Vaginal microbiome and NGS.
Yes. Okay.
Great.
Okay, so the NGS actually stands for Next Generation Sequencing. And I'm not a scientist or a geneticist, so we're not going to go into too much detail about that. But I think it's an important topic because we need to understand what's actually going on in there. I just want to go through a couple of things that we're going to be discussing. So we'll go through some definitions so you understand what I'm talking about. I also want to just discuss what a normal vaginal microbiome is and what can affect that so that we know what to do when things go wrong. And
Tina,
then we'll talk.
sorry, Tina, can I just say, I don't know if it's me, but I can see two slides. I can see your outline slide and then the next slide. Is that right, Chris? Or am I seeing too much there?
Yes, yeah, I was just about to say, maybe we can change that at display settings on the top there.
Let's just say...
I don't mind at all. It's always nice to know what's coming next. But I think you don't want to you were going to keep your powder dry. You don't want to steal your thunder.
Swap presenter view and slideshow, maybe.
Is that right, Steve? I mean, Chris?
I don't think that's going to. No, I don't think that's going to solve the problem. Oh,
there we go. You done it. Tina, I don't know what you pressed, but we got it.
I don't know either. Yeah, okay. If that works for you, then that's good. Okay.
That's perfect. Now we can see nicely.
Okay, great. Because I've actually got two screens open. So maybe the one, was for me and the one was for you.
Okay, you were sharing, but I think you were oversharing, as it were.
Okay.
We've got to have boundaries, yeah?
Okay. So NGS, as I said, was next generation sequencing, and I will discuss what that means in a second. So there are two words. One of them is microbiome, and the other one is microbiota, and those two are actually used interchangeably quite a lot. But if you look at the actual definitions, when we talk about the microbiome, that's usually refers to the collective genetic material. And if we're talking about a specific community of microorganisms, and specifically the vagina, it's actually got a lot of bacteria there. We've got fungal organisms like Candida, which actually should be there, that can actually overgrow. We've got viruses in there like bacteriophages and archaea. So we've got little parasites that live there as well. And that can all be actually completely normal in that specific environment.
Sorry, Tina. Tina, can you just say what is archaea?
So archaea is just another group of organisms. And they, so we've got the way that microbiology or microbiologists name organisms. They group them based on their, we've got genus and species and different, basically based on a taxonomical speciation and grouping. So archaea are it's another group of organisms, and generally you can have parasites that fall into that category.
Okay.
So the grouping, yeah.
Okay.
Yeah. So when we talk about microbiota, so if we're talking about the actual organisms and not the genetic material of these, so when we're talking about the actual organisms, we should be referring to them as microbiota, but a lot of people use the term microbiome. So if we want to be truly correct, then we have to use biome for the genetic material and biota for the actual bugs.
Okay.
And when there is an imbalance in this community or any community, and that can obviously be in an environment, in a lake, in any system, then things can go wrong. So that's what we're going to be discussing today. So in order to study various communities, we look at the DNA, so that genetic material. And in the past, we used to use a technique called Sanger sequencing, which I will discuss just now. But obviously, things will progress and technology progresses. And, we need next generation things. So the new techniques are called next generation sequencing. And essentially, this is a technology that helps us sequence, so basically put into order and analyze the building blocks, which are of the DNA and the RNA. So as we know, DNA and RNA are made up of these building blocks. So for DNA, we've got adenine, thymine, cytosine, and guanine. And then RNA uses the same bases, except it replaces the thymine with the uracil. And what the difference is with the next generation sequencing is instead of reading just one single strand of this genetic material at a time, it's able to essentially break down the genetic material into millions of small pieces, and we can read them all at the same time. So it has the power to essentially take an entire you can take it from soil, you can take it from water, you can take it from the gut or from the vagina, and we can basically map entire genomes in a single day, because you can basically sequence, analyze, and read millions and millions and millions of data that we weren't able to do previously. So this is just showing you the building blocks of the DNA and the RNA, and then just very, very basic steps of the next generation sequencing was obviously you need to extract the genetic material from the sample, whatever that sample may be. It gets the strands get separated and fragmented, and you have tags attached to it. And then it's sequenced. So essentially, you have to align it. And then you need quite advanced IT and AI tools in order to analyze this data or these data in order to make sense of it. And I will show you an example of that now. So this is also just another example of the work stages. So, you basically prepare these sequence libraries where you break the DNA or the RNA into little fragments, and then you're able to then sequence and basically align those into like a little string of beads that you can then analyze, and you can analyze them and compare it to a known sequence so that you're able to then tell, is this an E. coli or a Staph aureus, and how much of it there is by interpreting these data. So this was an example. It was actually an example of a couple of samples taken from patients with bacterial vaginosis in the vagina or from the vagina. And the vertical columns is one patient, and then the horizontal rows are essentially the organisms that they found. And essentially it shows you all the various bacteria on the right-hand side, and the red squares, so the more red there is, the more that organism was actually detected in that particular sample. And it just shows you, the abundance of the organisms that was found in that specific sample. And obviously then you can compare the various microbiomes to one another by looking at that. So
Tina,
yes.
so that huge list on the right-hand side of your slide there, those are
Yes.
all the organisms that were present
Yes.
in this particular individual's vagina.
Exactly. And remember, so this is a lot more sensitive if you think about what we are able to do from a culture perspective. A lot of these organisms are extremely fragile. They may be present in extremely low numbers. They may have very specific culture requirements, and we may not even be able to grow them. And obviously this brings into question, how many of them are actually significant in the pathologies that we see. How many of them are part of the normal flora? How many of them are real pathogens? How many of them should be there? How many of them shouldn't be there? How many of them are new that we've never seen before? And you can see one of them on that right hand side. I know it's very small, but some of them are, one of them is called unclassified fungi, which we've never seen, an unclassified bacterium. How do we know that that's something that may or may not be important in the sample of a patient who's got bacterial vaginosis? So, it's and one of the points that I'm going to make with next generation sequencing in any sample at this stage is, yes, we've got this incredible technology, and yes, we've got all these data, but how do we then tie that into treatment modalities, diagnostic modalities? How do we use these data and use it clinically, and how relevant is it at this point if we still don't know how to use it?
So that is the importance at the moment. If I say to a woman she's got chloroplasts in her vagina, it may or may not be significant, because these things may be very fastidious and not even able to be cultured. And we don't know, is it a good or a bad thing to have chloroplasts?
And we don't know. We don't know at this point. So I think there's still a lot of work to be done. So, I think, but we're getting there. We're collecting the data, and hopefully AI will help us with this. So what we'll need to do is obviously map this together with patient symptoms, and then perhaps correlate what organisms in abundance or in their lack would be related to patient symptoms. So that would be very interesting to see.
Yeah.
What we do know, and this is mainly from obviously culture-based studies, is that a normal vaginal microbiome is dominated by lactobacillus, and that usually makes up about 70 to 90% of the vaginal microbiome. And then Bifidobacterium comes in second, and it's normally the longum species and the breve species. In terms of the lactobacillus, we've got the many different species like Crispatus and Acidophilus and Rhamnosus. And I think the most important thing, and I have mentioned this in my previous talk, was that they produce hydrogen peroxide and lactic acid. And the main, the main pro is that it lowers the vaginal acidity, and we know that that's extremely important to keep pathogens out, to prevent overgrowth of candida, to maintain the integrity of the lining, and very, very unlike the gut microbiota, the vaginal microbiome is very low in diversity. But you can see that because it's populated mainly by lactobacilli. And when the lab sends you a report saying that the patient has BV, that usually means that there's a lot of E. coli and Mobiluncus and all sorts of other funny things growing because of the fact that there's low lactobacilli. So that's a bad thing. And the normal, remember, if I do let's say we do an NGS sequencing on me, my mom, some ladies at work, we'll all be normal and asymptomatic, but we'll all still have major varieties in our normal vaginal microbiota, because these things vary quite a lot by ethnicity, by age, by the hormones, what stage of our cycles we're in, our health status, whether we smoke or not, what diet we have, genetics, etc., etc. So it is still like a fingerprint. It's extremely unique. And once again, it'll differ whether it's a cervical swab, a vaginal canal swab, or whether you've taken it from the uterus. So, a lot of these things need to be considered as well. So what affects these microbiotes or the microbiome? Well, I have mentioned ethnicity is important. So they have looked at various studies. So they've looked at the Asian population, the Black population, the Caucasian population, and they've certainly found that in the Black population, the lactobacilli tend to be less predominant. So there is a greater a preponderance of BV. And also the lactobacilli species tends to differ between the Asian population, the Caucasian population, and the Black population. So that's also very interesting. With age, we generally lose lactobacilli, and we notice that as practitioners, because we see patients coming in with more candida as we get older. We see patients coming in with gut issues as we get older, because if we lose lactobacilli, you get constipation because you're losing lactobacilli and bifidobacteria in your gut, which affects your vagina as well. So we know that. Hormones play a massive role. We do know that with lower estrogens, your lactobacilli can drop, and you can have an overgrowth of candida, so hormonal fluctuations, whether it's perimenopausal, postmenopausal, during your period, certainly does affect the microbiota, which can also then affect, your risk of BV, your risk of candidiasis, etc., how healthy you are in general. Sexual activity does affect it as well. You can be of greater risk for bacterial vaginosis. Any sort of douching, because you're going to rinse out all the good bugs. So, generally douching is not really recommended. Or anything, putting anything up there, fragrance washes and things. Your gut microbiome, and I'll show you just now, there are quite a few articles. Sorry, was there a question? No. If you've got an unhealthy gut, you are probably going to have an unhealthy perineal flora, and then you'll have an unhealthy vaginal flora. So anybody who's presenting with recurrent vaginal discharge issues, we generally need to look at the gut microbiota as well. And I can promise you that they will have a problem with their gut as well. We know overuse of antibiotics will affect the microbiota all over. Probiotics can help, but it's not the only thing. Menstruation affects your microbiota, your general lifestyle, and I'll go through that as well. Your type of underwear. So we find that cotton underwear helps with breathing, prevents moisture buildup, increases the amount of lactobacilli, decreases the chances of candida overgrowth. So that's, the type of underwear sounds really ridiculous, but it certainly does help. Your overall immune status, and obviously overgrowth of STIs will affect your microbiota as well. So, there's a lot of different things to consider when you're looking at the overall vaginal health. So when your lactobacilli levels drop, you are at risk for candidiasis. You certainly are at risk for preterm delivery. We know that patients with BV are at risk for preterm rupture of membranes. The vaginal microbiome and the urinary microbiome are very closely linked. And once again linked to the gut as well. So, I heard Dr. Young speaking about urinary incontinence and all of that. So, so microbiota are very closely related. If you've got generally with patients who've got vaginal discharges are very often have recurrent urinary tract infections as well. you've got an increased risk for STIs, for BV, and for pelvic inflammatory diseases. And if your lactobacilli are low, more than likely you've got increased levels of Gardnerella, which we know is the main bacteria of vaginosis. We've got the growth of anaerobes, Prevotella, Epitobium, and all the other horrid little ones. So just to show you an idea of the organism, so this was actually NGS sequencing, just looking at, okay, let's take a group of women, do their BV scores, do the Nugent scores. We'll look at the normal group, we'll look at the intermediate group in the middle, and we'll look at the patients who've got obvious vaginitis. And they wanted to look at, okay, well, what organisms predominate? So if you look at the women who don't have BV on the left, you can see that the most predominant organisms, so 83% of their microbiota was actually lactobacillus. And you can see they had a couple of other things, and about 1.5% was Candida. So yes, Candida can actually and is often there. However, if you look at the vaginitis group, their lactobacilli levels were really low. It was 38%. And what was interesting is their Prevotella, which is typically associated with bacterial vaginosis, shot up to 27%. And then you've got all these other anaerobic organisms like Peptinophis, I can't even pronounce it, Mobiluncus, and other bugs, and Epitobium, which can also be associated with BV. So,
Tina, can you just, you mentioned a word, someone says, can you just reconstitute the concept of a Nugent score? And how does that work? What's a good one and a bad one, and what do you actually score?
Yeah, absolutely. So when we look at a BV score, essentially what we look at is the epithelial cells that you've sent us from the vaginal canal. So the most important thing would be the number of lactobacilli. So how many there are. I don't know the scoring system off by heart, but essentially it's the number of lactobacilli. So it's obviously moderate, medium, or low. The other thing is also looking at the epithelial cells to essentially see whether they are stippled with anaerobic bacteria or not. And the gram negatives that also sit on those epithelial cells, and that's also ranked to see whether or not there are a lot of them sitting on there, or not that many, or a few. So those are basically called the clue cells. There are various different scoring systems that different people use. The criteria differ slightly, but generally, the most important two would be the stippling of the epithelial cells as well as the lactobacilli.
Okay, thank you.
Okay, so if you look at the possible clinical applications of the next generation sequencing, we could then obviously, hopefully in the future, once we've got the patient info as well, we could possibly look at it for treatment plan formulation. We could look at then, how do we monitor those patients on the treatments? Remember, because we've got the whole genome of the patient, we could look at the resistance of the organisms as well. Will they possibly be resistant to the treatment that we are offering? And obviously we can sequence them and look at what the organisms are, how much is in there, which ones are significant, which ones aren't significant, et cetera, et cetera. Okay, so although it certainly can be a tool, and yes, it's comprehensive, and yes, the results are not affected by reader variability. Because obviously, if we're doing things manually, then one reader can vary from another reader, and certainly can correlate quite well with your MCNS. It's the interpretation is still not standardized. There's a lot we still need to learn. Will it be costly? Will it not be costly? Will the cost come down? We've got all this information. How do we know what to do with it all? How will we interpret all of this information? It'll certainly get better, and I think it's going to happen relatively quickly. There's also, from the articles that I've read, is they may even be able to predict vaginitis. with certain changes in your microbiota to say, okay, no, this patient is at risk and will be, there's some indicators showing that you may get vaginitis soon. And also some biomarkers that may say, there's inflammation and resistance genes, especially for the organisms that we can't actually grow. So things like chlamydia, mycoplasma, which we actually aren't able to grow because they're just so finicky, and they don't have cell walls. So that would be very, very helpful. So certainly, we are looking at it for the future. We just need to know a little bit more. So the last bit of my talk, I just want to touch on the importance of this axis. And I think, not only gut vagina, but gut, perineum, urinary, vagina, because if it's poor down there, it's poor everywhere. And there are a lot of articles kind of detecting this, and they can certainly prove that there's this movement between, poor gut microbiome. If you've got bad E. coli sitting there, you've got this migration of organisms from one area to the other, and affecting a continuum essentially, which certainly makes sense. And I think there's different components that they mentioned here. So certainly microbial migration from the rectum to the perineum and seeding to the vaginal niche, which can clinically obviously cause aerobic vaginitis, which I did mention in my previous talk, with an overgrowth of E. coli and Klebsiella, which can certainly cause inflammatory vaginitis, bacterial vaginitis, urinary tract infections, urogenital infections that are not STD related. So that's important. Also remember, the gut is responsible for a lot of conjugation or deconjugation of estrogen. So what about the hormone regulation? Clinically relevant, cycle shifts in that area. How does the gut affect that? How will it affect the vagina as well? Also a metabolite exchange. So we know that if the gut doesn't have the short chain fatty acids, which you'll get from your plant-based proteins, from your fibers, that can affect your barrier integrity and inflammation, and defense against your candida, and not only in your gut but in your vagina as well. So that's important as well. And we know about immune modulation. That's important as well in terms of your mucosal defense, your IgA and IgG balance with all your peptides, and that would be not only in your gut but in your vagina. And obviously just keeping a homeostasis to manage local infections and reproductive infections. So, it's a very, very complex field that I don't think we give it enough attention. And then once again, as I have mentioned, poor vaginal microbiome is certainly linked to recurrent UTIs, and I think anybody with recurrent UTIs, we should definitely look at how healthy their vagina is, and I certainly, when I get some phone calls about a patient who has recurrent UTIs, I generally say, well, what's going on in their vagina? And let's see what's going on there, because they might have a BV that's misdiagnosed. They might have low lactobacilli. Let's see if we can sort that out. So it's definitely something to think about. I think we must forget about the silo of, okay, the patient that's come in with a UTI, or the patients come in with only a vaginal. These are continuum. It's a full continuum down there. We've got women have 3 holes, not just one to sort out.
Hang on, what's the the urethra? Is that the 3rd hole?
Urethra. Yeah, unless I'm counting wrong.
Urethra, yeah. Vagina, and anus.
Exactly.
Okay, thank you.
Yeah. So how do we improve the microbiota? Well, I think, vaginal douching, I think in general is a no-no, unless you've got a huge amounts of pus pouring out and we need to do it initially to get rid of the pus, then I think it's okay for a short period of time. Obviously, sexual partners, any harsh soaps with many fragrances and additives, smoking, obviously generally improving your gut microbiota, and that's another talk by itself.
Can you give us a bottom line? How would a person just said, how do you improve your gut microbiome? In short, yeah. How would you approach a bad?
I actually go into it in my next 2 slides. So
okay, thank you.
Yeah. So one of them actually is avoid chronic psychological stress. So stress has a massive impact on your gut microbiota. And it's quite profound actually. Stress and cortisol levels damage your microbiota and your gut massively with massive disturbances. And it affects your vaginal microbiota as well. So that's huge. A healthy, nutrient-rich diet with a low glycemic index. So that not only affects your gut, but your vagina as well. So generally, I think we definitely don't eat enough protein. With aging, we get massive sarcopenia, which is loss of muscle. With loss of muscle, we don't have a glucose sink. Which means that we have a problem with all the carbohydrates that we're eating. Muscles are extremely important as we age for longevity. I don't think we eat enough plant-based things. I think we eat too much sugar, too much processed food, too many additives. I think probiotics are a good thing, but remember, all of our guts are different. Some of us need them and do well on them. Some of us don't. So it's not a kind of a magic bullet for all. I think we need to try and see. It may work, it may not. Sometimes it can worsen things. Probiotics, I do use a lot of raw probiotics and live for treatment of vaginal infections because it lowers the pH. So those of you who would have phoned me with chronic vaginitis, I would have given you my little protocols. So that is an option. But certainly it's very narrow spectrum for the gut. But that's another topic altogether.
Tina, you just go back one. We had a talk a year ago by a colorectal surgeon from Joburg, you probably know him, on gut microbiome. And he mentioned that you've got to eat 40 different types of foods every day. And that is, in other words, a nutrient-rich diet and a varied diet. You don't want to be a monophasic in what you eat. Would you agree with that?
Absolutely. The thing
Vegetables, fruits, legumes.
I think our diet needs to be varied. We get a lot of different microbiota from different things. I think we get a lot of different nutrients from different things. I always just say, just eat real food. Just eat things that we were supposed to eat like hundreds of years ago, that are not processed, that are not kind of man-made, that are not GMO, that are not. If we can just eat normal food, I think food in general just gets a bad name because it's because it's. I think if we all just stick to eating just real old school food, then it's fine. Then I think we can just eat as many different varied things as you possibly can, and as little processed additive shelf life food, and just eat fresh food.
How bad are takeaways and fast foods and junk foods?
The problem is that I think that the worst part about them is that the chicken that they buy will be the cheapest chicken that has grown the fastest with the growth hormones that have been injected so that the chicken grows like 10 times its size. It's been injected with growth hormones. It's been injected with antibiotics. It's been fried in oils like sunflower oil and canola oil. I just no, I disagree. Tomato sauce full of sugar. You get the point.
Get the point.
It's so from our side, I read a book about, health span, and, the diets have always been a bit tricky to unravel, and, there's never
They
really been
are.
found to be the perfect diet for our health. But they say that if you stick to the sides of the supermarket and avoid the middle, you'll probably be able to have better diet. So, the fruit and veg on the one side, good meats and healthy sort of proteins on the other side, and stay away from the middle where you got the biscuits and the chocolates and all that other stuff,
Exactly.
So, and
And as you
the other,
say,
yeah, Karen.
no, go ahead.
The other point is just that they also say that if your great grandmother wouldn't recognize it, don't eat it. And I mean, that's exactly the point you were raising, Tina. It's just good, solid, good food. So we can eat our
Absolutely.
meats. You can eat you can eat the most of the things. Because everyone wants to avoid carbohydrates, or they want to avoid this or that and the next thing. But as long as it's real food, eat it.
Yeah, I just believe in real food that's not processed, and the thing is, we will never know which diet is good for you because it's so difficult to study diets. It's really, really difficult because it's we are all so different. We've got different hormones, we've got different ages, it's really difficult to study a diet. So yeah, anyway, that's, I think, just a simple old school broad. Anyway,
Tina, your last thing, probiotics. What are natural probiotics? Are you talking about Bulgarian yogurt, or do you talk about some other form of natural probiotics that we can eat? Well,
I mean, you could certainly try. Some people don't do well with that because, when I give my gut microbiome talks, I always, kind of compare it to like a bag of Smarties. So let's say that my gut microbiome, I've got a whole bag of Smarties of all different colors, and let's say I've got a bit of a dysbiosis, and like most of my Smarties are red. Okay, and now let's say I'm eating kimchi, which has got probiotics, and kimchi's got lactobacillus in it, and it's got red in it. That lactobacillus, and I put more red into my already red Smarties mix. So I'm going to feel even worse and even more bloated. Whereas somebody else who's got mainly blues and greens, and then you put red into his, he'll start thriving because he doesn't have reds. So it all depends on the person. And remember, we've got thousands and thousands of different species. So adding, 3 different species from a kimchi or sauerkraut, although I think it's great, and I eat lots of kimchi, sauerkraut, Bulgarian yogurt, and all of that, and I feel amazing on them. And it makes me go to the loo regularly, which I find is amazing. And there's a lot of data on kefir and all of that. It may not work for everybody. So I think it's about trial and error,
Which food is rich in kimchi?
Sorry?
What is kimchi? I'm sorry. Is it like cream cheese?
No, no. Kimchi's like this Japanese, my husband thinks it's disgusting. It's like Japanese fermented rotten, it's rotten cabbage with like chili that the Japanese people make. Yeah.
That explains.
Yeah. It's kind of like a Japanese sauerkraut. Yeah, that's rotten.
Okay. Rotten Japanese sauerkraut.
Yeah. It's marvelous. Yeah. So here we go. Here's my overall microbiota health for the general. So getting enough sleep. So shift workers is a major issue. Working at night and shift work has a massive impact on your gut. That's quite concerning. And those of you who don't exercise, exercise is very good for your gut. And grandmothers always used to say, like, you must sleep at night, you must exercise during the day, you must get exposed to sunshine, vitamin D is good for you, exposure to nature, take your shoes off, walk through the forest, walk in the sand. Weirdly enough, or not weirdly enough, you'll actually feel better because your microbiome, and they've proven, actually improves. And we're all like, oh, those are such stupid things, but they actually work. So I know it sounds like very simple advice, but it's actually really true. So we need to go back to what our grandmothers did. yeah, and then obviously, as I said, avoid processed food and additives, avoid bleached water. So the one thing I never do is drink from the water in Cape Town because it smells of bleach. I think they do that so we don't all get cholera from water and salmonella. So yeah, I mean, yeah, unfortunately, you can't really drink the water from the taps, even though you won't die. It'll just, you're adding bleach into your poor little gut microbiome is not ideal. Avoiding toxins.
So the bleach, you mean chlorine? Is that what you mean?
Chlorine. Yeah.
Okay, so it's better to add, like I do, a tiny bit of whiskey to your water because that's a very good bleach.
Yeah, just avoiding toxins in general if you can. I mean, I've even read studies that taking like ibuprofens and Panados and blood pressure pills will affect your microbiome. But you can't avoid it. And we live in 2026. You can't like live in a bubble. You just have to live life. So you do what you can, and if you can avoid an unnecessary antibiotic because you've got a runny nose, then do that. And yeah, I mean, there's this interesting thing. It's called the disappearing microbiome theory. I don't know if you guys have heard it, but yeah, it's fascinating because they went into, I think it was the Amazon jungle, and they found this group of people, and they tested their microbiota, and they were like, oh my gosh. They compared it to the modern man, and they found so many organisms that literally just disappeared because we're so industrialized. And one of the bugs that a lot of people have lost is a bug that actually breaks down kidney stones. And what happened is that actually we're killing the organism that used to help us break down oxalates in our kidneys. So a lot of people don't have this organism in their bodies that helps us break down kidney stones. The other one is in our mouths that helps us with controlling blood pressure. So we're just losing these organisms that are, so what it used to do, it used to help vasodilate our blood vessels. So and now that we don't have it, obviously we vasoconstrict and our blood pressures will go up. So, we keep, because of industrialization, everything we're exposed to, we're just losing more and more bugs that used to actually help us. So, I mean, it's actually scary that we don't realize that we're becoming less and less diverse because we're just killing off all these little bugs that used to help us along the way. And they're finding it in these people that have never been exposed to modern society. So that's actually quite scary. Okay, I think this is my last slide. I did discuss a lot of products in my previous talk, so you guys are welcome to contact me. But the one thing that I actually thought about the other day, just because of the physiology of bacterial vaginosis, and there's this massive role of biofilm, and as you know, that's kind of like a jelly that the bugs sit in. And obviously, if we just add Dalicin C or Metrogel, whatever, we often get recurrence. And I was thinking, well, why does that happen? And one of the reasons is because there's actually this biofilm that's actually sitting there. So the one thing that popped into my mind, and we use it quite a lot in the bone and joint infections, we use it for sinuses and whatever, and for wounds, because we know that biofilm sits there, is the stuff called hypochlorous acid. It sounds scary because it's an acid, but this does not affect any healthy cells whatsoever. It only kills bugs. And what's nice about it is it actually penetrates through biofilm. And actually, Philip Zinn and I were chatting because we had this lady who had recurrent BV, and she's had a discharge. She actually allowed me to talk about her case. I think she's had a discharge for 7 years, or 5 to 7 years. I don't have the exact duration. I'm not sure if Philip is actually on this talk, so thank you, Philip, for letting me share. But anyway, we started her on this hypochlorous acid, and we followed up with lactobacilli intravaginal, and we've put this on a tampon and put it in. And then she's actually gave us feedback. I think it's only been a week, but she, for the first time, has had no discharge after years. So I'm actually quite excited about this. And yeah, I think it'll be great if you guys could perhaps just try this. And I mean, please phone me, and I'll help you through if you've got any patients with recurrence, because I think this is something that would really work. I don't work for this company, so I don't get any money for this disclaimer. I don't. I just thought it would be a good idea for you guys. So we have to push boundaries in order to help patients and try something new if our standard is not working. So my details are at the bottom here. If you need to speak to me about any of your cases, please let me know. And then I can help you with some weird and wonderful things, because we need to try new things to help our patients, and obviously it needs to be safe. So all these things are safe that we try. So I'll obviously never let you do anything that's dodge. And that's all I have to say.
Thank you, Tina.
So much.
Thank you so much, Justyna. It's wonderful. Just before Steve pops up with some questions, I've got a few questions here from all the people on our webinar. Vaginal homeostasis. At the recent SASOG Congress, we noticed that there are lots and lots of products that treat recurrent UTIs by means of probiotics. So that is something that's come through. There's an osteo product for women that have chronic UTIs treated with probiotics. The other thing that you were talking about
on that point. Sorry,
yeah.
on that point. Those of us that see a lot of patients with recurrent urinary tract infections, I think we've overstated the available probiotics in terms of their real impact on recurrent UTIs. I have been being quite frustrated. In fact, this morning I was talking to one of my patients about this, and I'm just not sure we're targeting the right combination. I know once I spoke to you maybe about 5 years about this, Tina, and you actually said that we should use Probiflora 9, but which seems to, at one point, we felt was a good combination. But, I'm just not sure if we hitting the right combination. It just doesn't seem to be impacting like I wish they do. I was actually going to ask you what you thought about this, because, I think we are, and I think we've got some newer strategies. There's this new stuff called UT Pro Plus AF, xyloglycan and something else, and it actually prevents proliferation of the E. coli and other bacteria up in the gut. So which seems to be a slightly better option. So rather than increasing your probiotics, but rather reducing your pathogenic ones that are proliferating. But yeah, what is your thought on the whole probiotic thing, Tina?
Yeah. So remember, the urinary tract is not, the microbiota of the urinary tract is not lactobacilli predominant. So as we know, the organisms will come from the gut as well as the perineum and possibly the vagina. So we kind of need to look at obviously decreasing the bad organisms in the gut and the bad organisms in the perineum and the bad organisms in the vagina. So my approach would obviously be targeting those areas first and excluding poor microbiota there. It would be interesting to see how we could, so obviously all the strategies to improve microbiota first, excluding BV and all the other things and overgrowths in the vagina first, and then obviously targeting the gut is a little bit more difficult. I'd probably have to look and see the literature regarding specific products and how they would, which one would be the best in terms of prevention of UTI specifically. Because you'd have to have quite a strong organism to prevent overgrowth of, because the organisms kind of have to fight one another in terms of who's the boss. So you'd have to have quite a strong organism to fight against organisms, because remember, you'd have to look at pathogenic proteus, pathogenic Klebsiella, pathogenic enterococci, pathogenic E. coli, all the organisms that are responsible for UTIs, and there's a lot of them. So, it has to be an organism or a number of organisms that have a significant impact. So I think it's a little bit more complicated than just giving a probiotic and hoping for the best. So I'd probably have to look at the literature, and have a look. But I'll have a look at that for you, Peter.
The trouble is, Pete, that some people are on quite a heavy, from a cost point of view. If you go for something like a Ruterina Fem or you get the multiflora women types that are in the fridge, and these things can cost up to 600 rand a month if you're going to be taking them. And these things are, these poor patients, they're really trying everything, to try.
Steve. Steve, on that, just to interrupt, I had my vet said I must do this probiotic for my dog, and I bought a pot of probiotics. It was cost about 68 rand for almost a kilogram of the stuff. And I think that, if probiotics are probiotics, how much badder can a probiotic for your dog be? Because is it not the same probiotic as one would use for human beings? And that's an interesting question, but I digress. Another person says, what about boric acid in the vagina? It's now available. Is it a good or a bad idea, Tina?
I think it is a good idea. I know the product you're talking about. It's got a very sexy name. It's I think so. What it never used to be available, and we used to have to ask the compounding pharmacy to mix it up. And the dose that they're giving seems to be fine. We generally use it actually for Candida glabrata because we had no treatment options, and I think that's great. Candida glabrata, I think it's a great product. Yeah.
Okay, that's good. Something that's slightly off-piste, but you're a geneticist and a microbiologist and a pathologist. They said sometimes the Pap results can nowadays show you whether or not a woman has chlamydia or BV or Gardnerella or other organisms in the vagina on the Pap smear. Why can't they find syphilis and HIV on Pap smear if they can find the others? Do you have an answer, quick, a 1 minute answer to that?
Well, you could probably do a PCR, but yeah, HIV I wouldn't really do because that would be, and you don't really want to be diagnosed with floating HIV that's not really yours. And then you get an HIV diagnosis on your Pap smear that's not really yours.
Oh, you mean it could be your partner's?
Yeah.
Oh, gosh.
So you don't really want that diagnosis. And syphilis, you're not going to pay 1,200 Rand for a syphilis PCR on your Pap smear if you can just have a serology test that costs like 150 Rand. So it doesn't.
So it's not practical. And but another quick question: the possibility of inoculation of babies at birth when they're born by cesarean section with microbioma and microbiota of the type that you'd find in your stool, because in the olden days you'll remember you used to take your finger, stick it in the woman's anus, and then inoculate the baby's mouth with it, and that some people found that a horrible experience. Is it not available nowadays with your wonderful new techniques and technologies to have this done scientifically without damaging the baby and introducing herpes and other things that we used to do?
So I'm actually really glad you brought that up, because there is a technique to do that. So what is actually recommended, and they're doing this overseas, but obviously some people are freaking out about it, is they're using, so if a baby is born by cesarean section, they've definitely proven that they get skin microbiota covering them, as opposed to vaginal microbiota, because obviously if you come out through the vagina, then you get covered by perineal flora, which is actually the gut flora. It's actually so because obviously during that whole process you get gut. It's actually perineum and the mic. Anyway, so what they do is, and you can actually Google it. It's all over. You're supposed to actually put gauze into the vagina. I don't know how long you keep the gauze in. It basically soaks up the vaginal perineal microbiota until it's moist, and then you smear it all over the baby, into their nose, into their mouth, on their cheeks, all over. And then you keep it on for a while, and you do it for a couple of days. I don't know the exact protocol, but it's essentially what you're trying to do is mimic the kind of the birth canal process, just so that they can then develop the microbiome as if they've come out through the birth canal. And the point of this is because they've obviously followed these babies throughout life, and they find that the ones with the skin microbiome are more likely to get asthma, autoimmune diseases, diabetes, obesity, autism, etc., etc. It's obviously associations and not causal. It's very difficult to link it causally, but it's definitely something that is. I mean, I think the problem is, there'll always be that one child that may get a strep group B that has been picked up,
or herpes,
or herpes or whatever. So one can potentially. I mean, the thing is, if you had come out through the birth canal, you would have probably gotten it anyway. I mean, would you have tested? You probably would have. I mean, you do anyway. I mean, you'll probably do a strep group B anyway. You're going to do STD testing. You could still do that before you do the, the gauze in the vagina.
Inoculation.
You could still do that. So, I mean, I think it's a really feasible option if there's so many things that are beneficial. So I'm actually pro it. When my friend gave birth in in London, I said to her, please, please, I'm the godmother. And I said, please, can you do this? And she looked at me as if I was mad. So anyway, I
Or
tried.
maybe if you took a pink swab and a pretty little bottle or something, and then it wouldn't be so objectionable. Now, the final question, I don't know if Steve's got any questions. And another one that's come up, there's been a sort of resurgence of products. Steve was talking about probiotics and things. Estriol and lactobacillus, the product we mentioned last time, you remember, is called Gynoflor. That seems to be something that has theoretical basis. Estrogen plus the probiotics in menopausal women does make sense, yeah.
It certainly makes sense because the estrogen is the reason why the lactobacilli are low. And we know that estrogen does help with the barrier and the epithelial integrity. So it does make sense. Yeah.
And estriol is going to be available very shortly through Allstar. Steve, I hear you wanting to say something.
Yeah, look, I've also been prescribing the Gynoflor. It's just quite a big tablet, eh? And patients are complaining a bit about the
But
size
it's
of
in the vagina. It's in the vagina.
Yeah, but some of the ladies do find it a bit uncomfortable to put in it so big, especially those with a lot of atrophy. So that is just something that they do complain
How big
about.
is it?
It's big enough that patient told me this morning she has to break it into pieces to get it in. So yeah,
Good heavens.
that's just a little practical thing. I mean, it's a great product.
Quite big as well.
Sorry?
The Polygynex is quite big. I know we had a young girl.
It's huge. It is.
And the mother was like, oh my gosh, I don't know how I'm going to get this in. It's so big. I didn't realize they were so big. Yeah.
No, they are big. These tablets are quite big, but yeah, I mean, just there's a group in the US who are also looking at the genetics of some of the microorganisms in terms of how they're taking an approach to treating patients with interstitial cystitis with a long-term antibiotic protocol. Ruth Chris is someone who's been doing this. And they are actually offering patients a commercially available urine type PCR type tests to see how to target this. So this is a trend in the US. I looked at it once in South Africa. It wasn't really feasible, something that we could do yet. But are there any thoughts on that, Tina?
I mean, we know that the urinary the bladder is actually not sterile. If we had to sample it and do sequencing on it, we definitely grow bugs. I think we still don't entirely know what they mean. And an inflamed bladder certainly has different bugs to a normal bladder.
Yeah.
So it would be really interesting. I just wanted to ask you, Steve, do you guys ever use methenamine hippurate as a prophylaxis?
We don't have it available in South Africa.
Oh, really?
No, we've tried. We've it's great. I mean, it's got good evidence. It's available in Australia and the UK.
Maybe we need to,
yeah.
Maybe we need to speak to your pharmacists and try and get it through Section 21 or something.
I've got a patient whose husband is a pharmacist, quite a successful, busy pharmacist, and he tried as he may to try and get into South Africa, but it wasn't possible. Yeah. So I also tried to get it made up at one of the reconstituting pharmacists. They couldn't get the raw materials or whatever they need. So I've had patients who've had friends bring it in for them. But yeah, there is actually good data for Hiprex, and it would be great if it was available, but it's just something we don't have.
And do you prescribe Urimune a lot?
He does.
I do, yeah. I mean, I've got a little example on my desk here, so cool. I do use it. Yeah, good data. For anyone who doesn't know, it's a vaccine that you use for recurrent UTIs. They take a spray under the tongue over 3 months, and it's effective against E. coli, Klebsiella, Enterococcus, and I think Proteus. So it's got those 4 in it. And the data is good, and the results have been good, and no adverse events, just not covered by the medical aids, but it's about 4,000 Rand for this box. And I mean, one of the things that I was actually discussing again with another patient today was the repeat treatment, so we kind of almost 2 years in now, 2.5 years in from our first treatment. So does it make sense to you to give another course after about a year or so, Tina, or what is your thought on that?
I mean, with these things, generally there's the susceptibility comes from like this irritated bladder wall. If you don't have, like, I mean, so in men, if you don't have like a large prostate where you've got a reason why it's so if there's no cause or source really, a lot of the time it can actually be just that irritation. So you almost, if you get through that irritation, inflammation, and the barrier of the wall settles, then you can maybe see how they go. The problem is if the reason for the UTI is still there, or the recurrence is still there. So like an untreated diabetes or a severe immune deficiency or whatever, then it does make sense to continue. So it depends on the patient, I suppose.
Yeah. Well, that's why I'm using a lot of cystostats as well these days, hyaluronic acid installations, and they have worked very well actually. So I'm
Okay.
yeah. So, I always say to my patients, the bacteria have to jump through a whole lot of gates, and if one of them's open, you're going to get an infection. So that's why I always say you have to get a number of things. You have to get the microbiome right. You have to make sure that the atrophy sorted, the uroimmune, with the immunology. If they've got a residual, as you said, fix the prolapse or sort out whatever's causing them to have a high residual. So that's why, it does take quite a lot of thinking to get these patients sorted, and sometimes we still struggle. So it's a very important conversation. But
Absolutely.
but I think we have come to the end. It's 6:01 now, Pete. So it is time for us to end. Tina, as always, the conversations with you are always very informative. We really appreciate the time you take to come and talk to us. There's a lot happening in the world of microbiology and the vagina, and
always
the more we discuss it, the more we realize we don't know enough, and we're not using enough of the right strategies. But I think we are kind of getting a good idea as to where we should be looking, and it would be nice to see if the scientific community could be working a little bit harder to help us on this. Because there's a lot of speculation here, but we do appreciate and I think the genetics and AI, I think that's certainly a good place for us to be looking. So we are looking forward to that. So, but thanks so much, Pete. Thanks so much for your time as well, Chris.
everyone.
And, we'll see everyone next week again. So thanks so much.
Thanks so much. Thanks, Tina. All the best. Bye-bye.
Take care, everyone. Bye.
Thank you. Bye. Bye.